In this module, we will be learning about one of the most influential studies for studying single neurons in the visual system. As stated earlier, our visual pathway is laid out in what we call a retinotopic fashion. Basically, there's a map that tells us which specific area of our brain is likely to be activated if we stimulate a specific area in our retina. The areas that become activated is what we call the receptive fields. This is the area of the visual field within which it is possible for a visual stimulus to influence the firing of a given neuron. Now, which neurons? It just depends if it's moving, if it's static, if it's color, if it's moving slowly. And if you're not familiar with this, please go back to our previous module where we spoke about retinotopic and the magno and parvocellular layers. Please note that the activation of a specific photoreceptor cell will bring about the activation of a specific ganglion cell, and the activation of a specific ganglion cell will consequently activate specific neurons in the striate cortex. This is what we call the organized body, or the organized mapping of the visual pathway. In 1979, Whoville and Wiesel had an influential method, a technique, with which they studied single neurons in the visual system of laboratory animals. Their research subjects were cats and monkeys. However, their understanding and their discoveries led to revolutionary processes, and in fact, these individuals were the recipients of Nobel Prize for this particular study. The way they conducted the study was by, first, they would put the tip of a microelectrode near a single neuron in a part of the visual system under investigation. During the testing, eye movements were blocked by paralyzing the eye muscles, and the images on a screen in front of the subject were focused sharply on the retina by an adjustable lens. After, in the procedure, they identified the receptive field of the neuron. The receptive field of a visual neuron is the area of the visual field within which it is possible for a visual stimulus to influence the firing of that neuron. The final step in this study was to record the responses of the neuron to various simple stimuli within its receptive field in order to characterize the types of stimuli that most influence its activity. Then, the electrode is advanced slightly, and the entire process of identifying and characterizing the receptive field properties is repeated for another neuron, and then for another, and so on. This would give us an understanding of how the receptive fields are responding specifically to neurons that are located in a specific part of the retina. Because of retinotopy, we find that the four similarities of this influential field were actually seen at all three levels. And by three levels, we're talking about the retina, the thalamus, the geniculate, and the striate system. The four similarities that were observed in cats and monkeys through Whoville and Wiesel's revolutionary study is the first one that we see here, that the receptive fields of foveal areas were smaller than those in the periphery, which are of rods. This makes sense because our fovea is actually quite small, and it is consistent with what we see everywhere else in the brain, including the geniculate and the striate. The second similarity that was found in here was that neurons' receptive fields are circular in shape. We will talk about how this holds true with cats. However, later we found out that with humans, the receptive fields were not circular in shape. In fact, they were more rectangular. The third finding here is that neurons were monocular. That is to say that each neuron had a receptive field in one eye, but not the other. So as we look at certain neurons in our brain, we find that there are dedicated neurons for each eye, and they are not shared in any way, leading again to the retinotopy of our visual system. And the last is the finding that many neurons at each level had receptive fields with excitatory, meaning the neuron was firing, and within the same field we also had inhibitory, which means that the off part was firing. Let me explain this last point a little bit because it is important. When Whoville and Wiesel shone a spot of achromatic, meaning uncolored light, onto various parts of its receptive field of neuron in the retina, geniculate, and striate pathway, they discovered two different responses. The neuron responded with either on firing or off firing, depending on the location of the spot of light in the receptive field. That is, the neuron either displayed a burst of firing when the light was turned on, that's what we call on firing, or displayed an inhibition of firing when the light was turned on and a burst of firing when the light was turned off. The next slide will put things in perspective, as I have some pictures that would help you understand this better. In this picture we have an illustration of the receptive fields from the retina to the geniculate and striate system. As you already know, many cells have receptive fields with a center surrounding organization, and that's where they found that it was a circular shape. Here you can see the circular component, and within that same receptive field there are some on-center cells that are on, meaning they're firing whenever we are striking the center of that, and that would be specifically with which neurons we impact at the retina and the geniculate level. And then within that same receptive field we also have off parts that could be turned on while the other ones are off. And that's where we look at, again, having the on and off firing receptive fields in here. Again, the on center cells would be on and responding when there is a light that strikes the center of the receptive field, whereas the off center would be responding most when the light would strike the periphery of the receptive field. Please note that receptive fields are made up of hundreds of thousands, millions of neurons. And so our retina is impacted at a specific spot, and depending on where it is because of retinotopic, then it's going to activate specific areas of receptive fields. The on-center and off-center cells respond best to contrast, and so we find that if we were to have both areas, off and on, of a cell's receptive field illuminated together, there would be little reaction from the cell. I want you to put this in perspective, and I want you to think as to what this means right here. This has actually been used by the military, although it precedes the study, which was done in the 1970s and 80s. But we find that with soldiers that wear camouflage uniforms, the reason why they're not as easily spotted is because their uniform blends in with the environment. By blending in with the environment, then you have a constant firing of the entire visual field, and you are not able to discriminate. If you were to only have a green environment, and then there's a person with a red shirt within, then this would allow for an easier activation of the cell, and so a lot more reaction would be here, and so the enemy, in the case of war, would be able to spot a soldier who's wearing a bright red shirt in the middle of the desert or a green environment. Same thing here. You would have some component of the activation of the outside based on the inside. But if you have both turned on, then you're able to mask the individual, and this is because our eyes are not reacting as intensely because it blends in.